Good morning. Welcome you again to the class of molecular biology of first semester. And today I'll be doing DNA replication with you people, assuming the fact that in 11-12 you have studied the Watson and Crick DNA double helix. So we move on to these are the books that I have referred. In fact, I have already sent a study material to you people. I have referred Russell Genetics, Pierce, and Watson. And there is there are two pictures of Leninger's biochemistry. So this is the basic, very simplified structure of the DNA double helix, where we have a sugar phosphate backbone, as you can see. Where's my pointer? There is a sugar phosphate backbone. We have a sugar phosphate backbone, and we are having the nitrogenous bases protruding inward, and we have a double bond situation and a triple bond situation. Double bond occurs between adenine and thymine, and the triple bond is occurring between guanine and cytosine. In this case, adenine and guanine are the purine bases, and thymine and cytosine are the pyrimidine bases. If we look into a later picture, this is a more elaborate, beautiful picture. You can see that double helix moving in an anti-parallel direction. So this is a 5' to 3' direction. Just to recapitulate, why is it called 5' to 3' or 3' to 5'? If you look into the structure of this particular strand, the one I am now highlighting, you see the 5' phosphate is at one end and the 3' hydroxyl group is at the other end. Because there is 5' phosphate at one end and 3' hydroxyl at one end, so this direction is said as 5' to 3' direction. The other strand is anti-parallel to it, and as you can see, we have a 3' hydroxyl end at one end, and we have a 5' phosphate end at the other. So again, you can see the direction, it's 3' to 5'. So if this is 5' to 3', this direction will be 3' to 5'. And these are the, as I said, we have, this is the sugar phosphate backbone, and the nitrogenous bases are protruding inward, as I said in my earlier picture, where G pairs with C by 3 bonds and A pairs with T by 2 bonds. This is a pace filling model of DNA structure where the two grooves, so this is the minor groove and this is the major groove. The two grooves are being shown. The one helix is a rotation. As you can see, this is 10 base pair completes a 360-degree rotation. The length of 10 base pair is 3.4 nanometer, but let me be clear, this is the B form of DNA. So whatever quantification I'm doing or saying here is about the B form of DNA. And the distance between two consecutive bases is 0.34 nanometer. So we have a 5' end at one side and a 3' at the other anti-parallel strand. And again, we have a 3' end at one side and a 5' end at the other anti-parallel strand. Now coming to the concept of central dogma. By the fall of 1953, the working hypothesis that was adopted once the DNA was discovered was that chromosomal DNA functions as the template for RNA molecules, and these RNA molecules then move to the cytoplasm where they determine the arrangement of amino acids. In 1956, this pathway was famously referred to by Francis Crick as the central dogma. As you can see, there are arrows. The arrows indicate the transfer of genetic information. So the genetic information passes from DNA to RNA, and the information passes from RNA to protein. As you can see, there is a arrow encircling the DNA. What does it indicate? It indicates that the DNA is supplying its own template for replication. So it's an autocatalytic process. Next, from the DNA template, the RNA is being produced or synthesized. So this is a heterocatalytic process, and based on the information on the RNA, the protein is being synthesized. This is again a heterocatalytic process. Another thing that we should always keep in mind is that, see the chemical nature of the three constituents in the central dogma pathway. DNA is deoxyribonucleic acid, RNA is ribonucleic acid, and protein is nothing but amino acids. It's a chain of amino acids. DNA is chain of deoxyribonucleic acids, and RNA is chain of ribonucleic acids. Just to recapitulate, what is the difference between the deoxyribose and the ribose sugar? If we look into the 3' position of DNA, you see there is a 2' position. How do we understand the position of a sugar? This is the functional group O. Then 1, 2, 3, 4, 5th carbon. Okay. So this is the 2' position of DNA. And in the 2' position of DNA, as you can see, there is no oxygen. Had it been a ribose sugar, there would have been an oxygen molecule present there. Since this is a deoxyribose sugar, hence there is no oxygen present here. Hence it is a deoxyribonucleic acid. Now coming to, once it was established that, once Francis Crick established the fact in 1956 that information moves from DNA to RNA to protein, automatically the question came that DNA is synthesized on itself. It was hypothetically proposed by Watson and Crick that DNA follows the semi-conservative model. However, the proof for this model, that indeed the DNA follows the semi-conservative model of replication, came from the work of two young scientists, Matthew MESELSON and Franklin Stern in 1958. So they did an experiment where, this is the hypothetical picture of that experiment. As you can see, initially 3 models were proposed: the semi-conservative model, the conservative model, and the dispersive model, as you can see. In the semi-conservative model, as you can see here, the double helix of each daughter molecule. So if this is the parental generation, so these are the daughters. So you can see the double helix of each daughter molecule contains one strand from the original DNA molecule and one newly synthesized strand. So if we see the parental color to be pink, you can see that the daughter is having one pink strand which is acting as the template, and on that template a new blue strand is being synthesized. So this is semi-conservative because the new daughter molecule has one strand from its mother, the pink one, and the one it synthesized from itself based on the information of the template and the raw materials provided. Now coming to the conservative model. What you can see, what is the conservative model saying? The conservative model is saying that the parent DNA molecule is conserved. So if this is the parent, you can see this is totally conserved in this form. So this is the first generation. So the parental DNA molecule is conserved, and a single daughter double helix is produced consisting of newly synthesized strand. So this is a newly synthesized strand. There is no trace of the mother strand in this one. This is the conservative model. And finally, the dispersive model. In the dispersive replication model, as you can see, the daughter molecule consists of strands, each containing segments of both parental DNA, and newly formed DNA interspersed throughout the length. So you can see fragments of parental DNA. These are fragments of parental DNA, pink color. And we can see in between fragments of the newly synthesized DNA, which is in blue color. Now, this was a hypothetical, these were the three models that were hypothesized. And Watson and Crick said that the DNA replication probably follows a semi-conservative model, but there was no experimental proof to support their statement unless and until in 1958, Matthew MESELSON and Franklin Stahl went on to discover the semi-conservative model of DNA replication by their famous experiment of 14N, 15N. So we'll be studying that. So now we will be moving on to the MESELSON and Stahl's experiment. But prior to the starting of the experiment, let me just say the background of the experiment. They took the bacteria Escherichia coli for the experiment model. They grew the cells of Escherichia coli in ammonium chloride, where the nitrogen, sole source of nitrogen was 15N, which is the heavy isotope of nitrogen. As we discussed earlier in our class today, that one of the important components of DNA are the nitrogenous bases. So when E. coli started synthesizing DNA, it used 15N nitrogen as the source of nitrogen and started incorporating the 15N nitrogen into its DNA strand. Since 15N nitrogen is a heavier isotope of nitrogen, thus the density of the DNA with the 15N nitrogen in the nitrogenous bases thus increased. So this is how they could differentiate between a DNA having the normal nitrogen, that is 14N, which was a bit lighter, and the DNA having the 15N, which was a bit denser. So now let's move into the experiment. Can you hear the sound? So, as I said, preliminary, that it was hypothesized that there are three models of DNA replication: conservative, semi-conservative, and dispersive. We discussed this. In the semi-conservative model, the newly synthesized DNA strand is totally newly synthesized, and the another strand is conserved, that is a parental. As you can see in the semi-conservative mode, the two mother strands separate and a daughter is synthesized on the mother strand. And finally, so we have one strand daughter and one strand mother. Dispersive mode, as you can see, the mother strands are fragmented, and on the fragmented mother strands, based on the info, the newly daughter strands are then synthesized. So we have a mixture of both the mother and the daughter strands. So this was hypothesized. As you can see, they are showing it by the yellow highlights, that what are the three cases that were hypothesized. Now, coming to the experimental part. So this is, as you can see, the two young scientists, Matthew MESELSON and Franklin Stahl. And you can see the importance of nitrogen. They have shown the nitrogen bases. So this is the nitrogen. So where the E. coli were grown in heavier nitrogen, all this nitrogen is replaced by the 15N isotope. Now, as I said, they grew E. coli in a medium containing ammonium chloride, which was the sole source of nitrogen, and contain sole source of nitrogen, and the ammonium chloride had the heavy isotope of nitrogen. So if we grow it only in ammonium chloride 15N, it's heavy. When we have a heavy light and then a light. So see, the cells are grown in a cultured 15N for many generations. After many generations, the cells were taken from here and then grown in a vial containing 14N nitrogen. And as you can see, then the growth was monitored. Let me remind you here that the generation time of E. coli is 20 minutes. So after many generations, when they had grown here and the density of the DNA was isolated and checked, it was seen that the total DNA contained, it was a heavier DNA showing a heavier band. So the DNA contained all the nitrogenous bases, nitrogen having the 15N isotope. Now, after it was ensured that the E. coli is now having a heavy DNA, the E. coli cells were taken and grown in a cultured 14N. And now the time was monitored. After 20 minutes, some cells were taken, recovered, DNA isolated, and was tried to identify what kind of DNA was it. Was it the heavy DNA or the light DNA or a DNA having a heavy strand and a light strand, which is known as the intermediate form. So you see replication cycle 0. So that was the first cycle. After 20 minutes, they allowed the growth of the cells. They took some E. coli cells from here and allowed the growth of the cells in this medium for 20 minutes. And they took out the first batch of cells, which is known as replication cycle 0, to check the nature of the DNA, whether it was a heavy or a heavy light or a light. This is the second cycle. This is the third cycle. So after every 20 minutes, they took out the E. coli from the vial. Now, how were the DNA studied? As you can see, this is a centrifugation process where the DNA is isolated and subjected to centrifugation by cesium chloride. When you put cesium chloride in a test tube and the DNA in it and start rotating it, what happens? The cesium chloride arranges itself as per its molecular density gradient. So we have lightweight cesium chloride here and the heavier cesium chloride end here. Now, what is the logic of separating the DNA in this centrifuge tube? Was that the DNA will separate and stand at that level of cesium chloride where its density matches the density of the cesium chloride. So if we have a cesium chloride gradient of light to heavy, heavier cesium chloride on the bottom of the tube and the lighter cesium chloride at the rear end of the tube, I mean to say at the beginning of the tube, and when this DNA starts separating, so the heavier DNA will be towards the bottom of the centrifuge and the lighter DNA, if there is any, will be at the open end of the centrifuge tube. Further, if there is a DNA which is having a density of intermediate strength, that is light and heavy both, then that DNA will stand somewhere here. So the DNA is totally separated based on where its density matches the density of the cesium chloride gradient. So that was how the DNA was isolated. I'm going to say the density of the, based on the density of the DNA got segregated in the test tube. So as you can see, they're showing it in the picture. This is the heavy form of DNA. If there is an intermediate form, then we are seeing a band of intermediate form. And if in the DNA sample there is a light form, we are again seeing a band of the light form of DNA. So that was the experiment that was done here. So the three bands, as you saw, were observed in the cesium chloride density gradient centrifugation. Now let's see how they rejected the other two probable pathway of replication. So when we have replication cycle 1, as you can see in the lower side, they saw what did they see? In replication cycle 1, the result showed that there was a light density. Just let me go behind. The result showed, as you can see, that there is a light band and the result showed that there is an intermediate band. Below here is the result. The result showed that the DNA after replication cycle 1 showed a band of intermediate density. So this was the result. What was the hypothesis? Hypothesis would be that if the DNA followed the conservative mode of DNA replication, then one band would be light and one band would be heavy. As you can see, the pink one would have been the band with the heavy isotope, and the blue one, since it was cultured in 14N, the 14N vial showed a blue color. This was the light DNA. So the band would have been one heavy and one light. Two distinct bands would have been observed. But in reality, the experiment showed that there was only one intermediate band. So it proved that DNA does not follow the conservative pathway of replication. So from replication cycle 1, one thing was clear, that DNA is not following the conservative pathway of replication, as you can see. But there are two other pathways which are also showing DNA of intermediate density. In the semi-conservative mode, as you can see, we have a DNA of intermediate density. And in the dispersive mode also, as you can see, the DNA is showing, the daughters are showing a picture of intermediate density. So from replication cycle 1, it was not possible to understand whether the DNA is following a semi-conservative mode of replication or a dispersive mode of replication. So this was one thing. So the only thing from the first replication cycle that became evident was that the DNA is not following the conservative pathway of replication. So now we concentrate on these two pathways. Since the replication cycle 1 showed intermediate density, and as you hypothesized also, they are showing intermediate density. So we had to, we were not sure, I mean, they were not sure what would be the case. Now move on to replication cycle 2. Look at the result. The result is showing that there is one band of light density and one band of intermediate density. So this is the result. So for a second cycle of replication, when it was done, the result that was observed was there is one light band, that is the DNA contains only the N14 strands, and other was the intermediate band where the DNA contains a band of light strand. The band showed that the DNA contains one strand of light nitrogen and the other a heavy nitrogen. Now let's see how this happens. So now we are moving on to replication cycle 2. Look into this. Had the DNA followed a dispersive pathway of replication, let us assume, had the DNA followed a dispersive pathway of replication, then here also, in the next cycle, the DNA would fragment. We would be getting some original mother strands and we'll be getting some newly synthesized strands. Since the E. coli is now completely growing in a medium containing 14N nitrogen, hence the amount of 14N nitrogen would be more, no doubt of it. But still we would get intermediate bands. You see, there is not a single band where you are seeing, not a single DNA strand, sorry, not a band, not a single DNA double helix, you can, as you can see from the picture, not a single DNA duplex where you can see that both the strands contains 14N. it is still a mixture of 14N and 15N. Maybe 14N amount is more than 15N, but all the bands are showing a mixture of all the strands are showing a mixture of 14N and 15N. So there was no pure DNA strand giving us a pure 14N strand. But the result showed that there is a pure band of DNA containing 14N nitrogen only. So the next part, that is the dispersive model, was also rejected. This cannot hold true given that what was the result that we observed. So from the second cycle of replication, it became evident that since there is no pure since there is a pure band of DNA obtained containing N14 nitrogen in both the strands, obviously the dispersive model of DNA replication is not being followed. So ultimately what stays is the semi-conservative model. So let's look into it. Let's look into the animation. It's going a bit slow. So dispersive was rejected, and now coming to semi-conservative. So as you can see, when we have a second set of replication in the semi-conservative model, you can see the pink and the blue. In second replication cycle, the pink and the blue strands are separating. And on the blue strands, another blue strand is being synthesized because now N14 has been symbolized as blue color. So we are getting a pure strand of N14. We are getting a double-stranded DNA containing both the strands having N14, and we are getting a double-stranded DNA containing one strand, there's a pink one, having N15, and the blue one having N14. So if we now do density gradient of such a DNA, what will you observe? We will observe an intermediate band, and we will observe a pure band of N14, and that was what they observed in the actual experiment. So if so be the case, so the experiment proved the hypothesis which Watson and Crick had said, that most probably DNA replication follows a semi-conservative mode of replication. So as you can see, so in this case, we would be obtaining a band like this, and this is what they observed in the actual experiment. So the conservative model, as you saw, was rejected out. When we went for the two remaining hypotheses, as you can see in replication cycle 1, we obtained an intermediate band, which is seen here also, and an intermediate band, which is seen in the dispersive model also. So from cycle 1, it couldn't be decided whether it is a semi-conservative model or a dispersive model. Let's move on to cycle 2. From cycle 2, you can see via the hypothesis that via this, we can see we are having one pure band of N14 and one intermediate band, that is light DNA band and the intermediate DNA band. And here you can see the hypothesis that we will be getting two intermediate bands, even though the amount of N14 might have increased, but we will be getting two intermediate bands. And what did the experiment show? The experiment showed that DNA was obtained in two bands, one light band and one intermediate band. When this happened, it conclusively proved that the DNA replication follows the semi-conservative mode of DNA replication. And I was talking about this picture while I was showing the animation, that when they started, they ensured that both the strands contained N15 DNA, heavy DNA. Replication cycle 1, we get one strand heavy and one strand light. So it's intermediate band. replication cycle 2, you see one pure light DNA strand. So it's one pure light DNA strand, and see, this is the intermediate DNA strand. We are getting an intermediate DNA strand here, but the strength of the band has become lightened because since the amount of N14 has also increased in this intermediate band. And further as we move, we are seeing that the light N14 band is constantly increasing, but there is a faint presence of the intermediate band because there is only, if you remember, if you go through this picture, you can see there's only one DNA strand containing N15 that is remaining. All the newly synthesized DNA strands now contain N14. So once it was sure that DNA replication follows the semi-conservative pattern of replication. So there is one template strand, and on that template strand, a newly synthesized strand, a strand is newly synthesized. So automatically, the next question comes is that how does the phenomenon of replication takes place? So to get into the molecular concept of replication, we need to have an idea about what are the basic raw materials required for replication. So the foremost important raw material required for replication is the 4-deoxynucleoside triphosphate. This deoxy-GTP, deoxy-CTP, deoxy-ATP, and deoxy-TTP. So this is, where did my cursor go? So as you can see, this is a deoxynucleotide. This is the structure of a deoxynucleotide triphosphate, and the nitrogenous base. So this is the deoxynucleotide triphosphate, DNTPs, where N can be a C, a T, a G, and a A. So this is the first thing required. Second thing, if you look into the structure of this particular molecule, so the base can be anything. This 3', you can see this 3' is written in red, is very important. And there is an oxygen present in this 3' end, which is all the more important. Because this oxygen is very essential for the polymerization reaction. So this is one very important molecule that is essential for polymerization. And if you look into the 5' end, look into the other side, you can see there are 3 phosphates. This is the, so you can see there are 3 phosphates. The phosphate that is attached with the carbon is the alpha phosphate. The phosphate that is attached with the alpha phosphate is the beta. And the next one is the gamma phosphate. Usually when a trinucleoside phosphate is brought in, you can see, so this is the structure where a trinucleoside triphosphate is being brought in. So when it is being brought in, you can see where the cleavage is occurring. The cleavage is occurring between the alpha phosphate and the beta phosphate. So it is the alpha phosphate that gets attached to the protruding 3' oxygen. So that is how the polymerization actually occurs. I'll be showing you the polymerization by animation, but since we have a bit of a crunch of time today, we'll do that next day. So the first raw material that is required, as you can see, for DNA replication is deoxy-NTPs. Now coming to the second raw materials, the materials, as I'm saying plural, which becomes very essential for replication, is the primer template junction. So this is the primer template junction. The template strand is none other than the mother strand, as you can see. So this is the template strand. This is none other than the mother strand itself, which has opened up so that a daughter can be synthesized over it based on the information it produces. What is a primer? In case of in vivo DNA replication, it has been observed that if you just open up a DNA strand and give all the raw materials, replication will not start. It needs a small amount of RNA primer. So you can see if this is the mother strand, which is opened up, always remember this rule. The RNA primer, which is nothing but 5 to 10 nucleotide length, 5 to 10 nucleotide long. It's made up of ribonucleotides. The RNA molecule or the RNA primer always sits with its 5' end to the 3' end of the mother. That's the golden rule. So if you can see, where did the cursor again? If you can see here, so this is the 3' end of the mother strand, and the RNA primer is sitting with this 5' end on the 3' end of the DNA. Because you have to always remember that DNA is anti-parallel in nature. So the synthesis should also occur keeping the orientation of the DNA fixed. There is another thing that also should be kept in mind, that new nucleotides can only be added to the 3' end. New nucleotides cannot be added to the 5' end. It can never be done. I'll not say it can never be done. Let's keep that an exception. As per the normal rule of DNA polymerization, new nucleotides are always added to the 3' end. So keeping that new nucleotides are always added to the 3' end, then we have to, so the RNA has to keep its 3' end free. So it binds with the 3' end of the DNA with its own 5' end. So we have a stretch of small RNA primer that initially binds with the mother DNA at the 3' end. This primer is synthesized by an enzyme known as primase, which is a RNA polymerase dedicated to make short RNA primers of 5 to 10 nucleotide long on a single-stranded DNA template. The moment the primer is formed, now comes the formation of this area, which is known as the primer template junction. What is the function of this primer template junction? As I said, the primer provides the 3' OH, which is required to break the bond between the alpha and the beta phosphate. And what is the function of the template? The template guides what base is next going to come. If the mother has a G, then automatically the next base which is going to be attached is C. If the mother has a C, then the next base which has got to be attached, as you can see, is a G. So we have to keep this in mind, the importance of primer and template. The primer has an exposed 3' OH, which is used to break the bond between the alpha beta, and thus helps in addition of nucleotides. The template provides a single-stranded DNA that will direct the addition of each complementary deoxynucleotide. So these two basic things are required before or at the outset of replication. We need to have the 4 categories of DNTPs at hand, and we need to have this primer template junction ready so that the polymerization of DNA can move on. The polymerization of DNA or the addition of nucleotides is a critical process, and a single enzyme is unable to do it. So there are a host of enzymes which are involved in making this thing happen. It itself is a big topic, so I'm not starting that today. So we'll end here today, as to the basic two raw materials required, the DNTPs and the primer template junction to start the process of DNA polymerization.